Welcome back, everyone, to the Cannabis 360 conference. My name is Jason. I'm the manager of operations here at AEPS. I'll be your moderator for this panel on cannabis analytical testing. Nice to have you all join us today. We're going to be covering a lot of commonly asked cannabis analytical lab testing questions, and also to talk about some of the new trends that we should be looking out for. But before we dive in, we're just going to go through a quick introduction with the panel. A little bit about myself first. I come from a chemistry background with a little bit of analytical science experience as well. I worked in quality control and R&D laboratories under GMP regulations for a number of years prior to coming to AEPS. And at AEPS, I led a lot of in-house and corporate training relating to GPP, quality assurance, quality control, specifically for the cannabis industry. And for this panel, we have a very special guest joining us. We have the president and co-founder of Sigma Analytical Services, Ashton Abrahams, for this talk. We're very excited to have him here. Ashton, if you don't mind, before we begin, do you mind introducing yourself? Tell us a little bit about what you do over at Sigma Analytical Services, and how this panel, what we're going to talk about in terms of the cannabis industry and lab testing in particular.
Thank you, Jason. Hi, this is Ashton Abrahams. I'm the co-founder and president at Sigma Analytical Services. Sigma is a cannabis-focused GMP-certified lab headquartered in Toronto, Ontario, but we have international operations too, with a joint venture in Colombia and a few other countries we are active. We offer different types of services to cannabis industry, from testing to product development and formulation support, stability studies, cleaning validation studies, and different types of consulting services. As the cannabis industry, 3 years into legalization, it's still growing and expanding and evolving. So we are always facing new challenges and a new landscape. New questions arise, and we need to answer them. So I look forward to sharing some information on this panel, and I hope that the audience find it useful.
Fantastic. Thanks for that. And, we have a lot to cover, and I just want to dive right in. You did mention that, we are approaching 3 years into legalization of cannabis here in Canada. But, 3 years in on the analytical testing aspect, we're still hearing concerns about inconsistent lab results, for tests like potency testing, pesticides. How valid are these concerns, and, who should be addressing these?
There are many different factors affecting lab results. The very first one is how good your methods are and if and how they are validated. Cannabis testing here is a relatively young field, comparing to, for example, food or pharma. So the methods are not already completely established and available to everyone. And every lab had to work on developing their own methods for testing cannabinoids, terpenes, pesticides, mycotoxins, etc., in cannabis and derived products. So not all test methods are the same, not all of them are equal. And then it goes also to the instruments that the lab uses. Are they new, state-of-the-art, high sensitivity ones or not? Another factor is your team. Not every test and not every sample are just routine and regular, and different challenges arise. So it matters if your technical team has experienced chemists and microbiologists, with PhDs and master's degrees, or you have only hired based on whose salary expectation is lower. The process of testing, sample prep, data logging and review, and how you do it is, of course, important. Finally, it's about the consumables that the lab uses. This is another factor that affects the accuracy and reliability of your results. And most importantly, among those consumables is the standards that the labs use. So it's not only about if you buy the highest quality standards or the cheapest ones, but it's also about how often you do your standard prep. Because standards degrade after preparation. And if a lab keeps using the same one for a longer time to save some money, then it would affect the accuracy of the results that the lab is reporting.
Right.
So there are many factors potentially affecting the results and their accuracy. If you send the same sample to the very few labs who are doing a good job right now, covering the factors that I listed, you will get similar results from them. But unfortunately, 3 years into legalization, there are still many other labs who are sacrificing accuracy of results to cut the costs. This is a problem for which not only the labs, but also the LPs pay the price, because look, inaccurate results could authorized release of a batch that would fail upon Health Canada's audit or retest, and end up with a recall and significant loss to the LP's bottom line and also their reputation. So the LPs need to be careful about picking their labs, and their quality division should do a full qualification of the labs to pick one whose results could be trusted.
Yeah, no, and you actually unpacked quite a bit there. Really, you mentioned standards, validating methods, sampling procedures. And I think 3 years in, is it should Health Canada really be sort of stepping in and having set guidelines on some of these validated methods to really help not only the license holders but also contract labs to ensure that there's consistency between from one lab to another and the way that they test. And I know each lab has to do their own validation, but ultimately, this responsibility of having consistent, results, who can really, guide the ship, just like we have in pharma and food, where no matter which lab you go to, the results are usually within, 1 or 2 percentage points within one another, right? So on the cannabis side, why are we still seeing sort of inconsistencies? I guess what our audience would probably want to know.
There are two ways to control this. Of course, one way is what you mentioned. Health Canada needs to do proficiency tests, or keep a closer eye on what labs are doing and their validation methods. They are doing things. We are seeing it, especially recently. Some of our clients got audited, and we noticed that one of the documents that Health Canada asked for was the validation reports from the lab that did the testing for them. So I'm not saying Health Canada is not doing anything. Of course, they are doing their job, but they can do better and they can do more. And I think they're on track to get there. But until we get there, I think it also falls on the shoulder of the LPs. They should be careful, and they should just do a full qualification and make sure that the lab will provide them with accurate and reliable results.
That's right. And of course, they should be approaching only labs with analytical testing licenses for cannabis. And, in terms of, I guess, LPs from their perspective, we when we talk with license holders and clients, and there's still a lot of misinformation about labs and sort of getting the desired results, higher THC or lower contaminants or whatever the case may be. So they sort of lab shop around. So why do you think this is still happening? Is it just a matter of sort of education, or a matter of better understanding of where lab results come from? Why do you think LPs are still doing lab shopping for better results?
In terms of lab shopping, we see two problematic approaches in the market, when LPs choose their labs. One is disregarding accuracy and reliability of the results and qualifying their lab, and simply make their decisions based on the price, as I told you. Because if you look at what I listed for the factors which affect accuracy of the results, you see each and every one of them are costly. So quality is costly. When a lab's competitive advantage is simply lower prices, they could not have achieved it unless they just saved on the cost of standards, instruments, personal review process, therefore sacrificing the accuracy of the results. So unfortunately, we still see it in the market among the LPs that when they just go look at different labs and they want to pick the right lab, that's what they look at. Surprisingly, I had a call with a senior manager at one of the largest LPs in Canada, a very well-known name in the industry. And when I asked him if he is going to ask the quality team to qualify our lab, he said, this is not how they do it. He said they make the decisions only based on the lab's price list and nothing else, which was surprising to me. Another issue is exactly what you brought up. Another problematic approach in choosing the lab is the LP's target here is to get the highest results for cannabinoids and lowest results for contaminants instead of the most accurate ones. This is very risky to the consumers, as it means they cannot trust the safety and efficacy of the product, and also to the LPs, as they take the risk of not only losing their market trust, but also facing a potential recall. So those LPs basically risk millions of dollars to save a few $1,000s. But the good news is that with Cannabis 2.0 products gaining higher market share and also becoming more and more diverse, we are seeing this harmful practice of lab shopping less and less, because as here, higher THC content is not an advantage anymore. And for these cases, the LPs only want to hit their potency targets.
Right. No, that's great to hear that, with Cannabis 2.0 actually has reduced some of this lab shopping practice. Coming from a analytical background, it was kind of quite shocking to hear in sort of the first 1 or 2 years of legalization. And this ties sort of to the first point, right? Once Health Canada dedicates more resources into auditing validated procedures, we'll then see a bit more consistency between labs. So then, lab shopping wouldn't really affect or impact results as much. But no, those are great points. And speaking of like variations between labs, when we do consulting with clients, we see all sorts of certificate of analysis. So for those of you in the audience who may not be familiar with that term, CFA, basically it's a summary sheet that talks about the results of each and every single test that's done on your sample. And we see all forms of CFAs all across Canada from different labs. Can you clarify for us, Ashton, what should be included on the CFA? What is it, various sort of information that's needed or required that Health Canada wants to see, and also your clients will want to see?
Sure, there are clear requirements for what needs to go on the CFA in the regulations, but only for reporting pesticides. That's the only one. In addition to some information about the lab and the person requesting the analysis, and the sample, there also needs to be information about the method, instrument used for testing, LOQs, limits of quantification, attestation and signature, and etc. But the requirements are not as clear for the other tests. Mm-hmm. I personally have not seen it in many other cannabis labs, but here at Sigma, what we do is that we include almost all that goes on a pesticide CFA for all the other tests too. So in addition to that information about the test and instrument and the sample, we also add some supplementary information that we have on our CFAs, which are not required, but the client finds them helpful. For example, a list and graph of the dominant terpenes, or a chart of the major cannabinoids in the sample. But you're absolutely right, there is not a standard practice here, and those bare minimums only apply to pesticides. For the other tests, they are voluntary. But I think it would be beneficial to the whole market, from the LPs to the labs, if all labs start this practice and add those informations on each and every CFA, even if it's not for pesticides.
Mm-hmm, mm-hmm. No, those are great points. And one of the and we'll talk about sort of other I guess non-required tests later on in our panel today. But recently, Health Canada, with at least some of the clients that we were dealing with, they're a little bit more focused on justifying the specifications, the specs, depending on the intended use. So depending whether the product is ingested or inhaled, there's different limits and different specifications, right? Yes. So the question that we always get asked is, should the contract lab be providing the specifications, or should this be left, to the client because they know best what the intended use are for the products?
Look, for any contamination, whether it's microbial, pesticides, heavy metal, mycotoxins, the specification is on our CFA, but how is it picked? Let me explain also something. For profiling cannabinoids and terpenes, it could be report only or for information only. So here is where you do not necessarily need to have a specifications. But about what the specs need to be, there is no quick and simple answer. It's not that simple. It's a little bit complicated, because Health Canada has established some of those specifications very clearly. For some others, they have referred to pharmacopoeia. But again, you have more than one option to pick from, because you have, USP, you have European pharmacopoeia. Even in each pharmacopoeia, you have different options. Yeah. They also depend on the product type and intended use. You made a good example here. For example, is it for digestion or inhalation? Of course, then the specifications will be different. And still, in some cases, the LPs have some flexibility to pick them. But to give a clear answer to your question, the specifications must come from the client and are not a lab's job or discretion. Here at Sigma, we offer our advice and recommendation if a client asks for it, but at the end, the client is in charge of picking the right specifications. Right.
So basically the QA of the license holders should have the knowledge and foundation to know what the specifications is, which pharmacopoeia to pick from, whether it's for inhalation or ingestion. That's based solely on their decision. Is that correct?
Yes, and keep something in mind. At some point, either later they get audited, or if they just simply want to release a batch of their products, and Health Canada looks at the certificate of analysis, only the LP is in charge of the specifications there, because they have chosen it. So
Right.
if Health Canada is not happy with them, if they think that the wrong specifications have been chosen, they go to the LP and question them, not to
That's
the
great.
lab.
No, that's great to know. And one good point that you actually brought up is differences in methodology, differences between just because in part of the cannabis regulations they still refer you to Schedule B of the FDA, and oftentimes I get asked, is it the Food and Drug Administration? No, it's the Food and Drugs Act in Canada, which refers to a list of pharmacopoeias. So you have British, European, you have United States pharmacopoeia, and of course in those pharmacopoeia there isn't a specific chapter on cannabis because it's not recognized as a medicinal product yet at this point in those jurisdictions. And this leads to a lot of variability in terms of testing. In particular, for example, microbiology contamination, right? Microbials. Which methods are reliable are actually approved by Health Canada, just because it's slightly different from USP versus EP, right? So what's your sort of experience and take on that?
You're absolutely right, because the whole legalization of cannabis is new. The pharmacopoeia has not yet caught up with it. So you need to either look at herbal medicine or pharmaceuticals, and there is nothing very clear here to do about that. But keep something in mind when you talk about that. Methods and technologies and pharmacopoeia, it goes above and beyond simply the specifications. Because when you look at the method, it's not only limited to what's in USP or what's in European pharmacopoeia or British pharmacopoeia. On one hand, you're right, because Health Canada has just been flexible here and has left it to the LP to pick the right one. On the other hand, it goes above and beyond, because the whole field of testing for microbial contamination is a very dynamic and evolving and fast-growing one. So it's also about the different technologies which are being used, not simply those standards. And there are different methodologies, different technologies to test for microbial contamination, from the traditional method of plating to newer and accelerated technologies like Petri films and Qpcr, which could all be used for testing microbial contamination. What we recently are seeing in this regard is really unfortunate and disappointing, because labs are supposed to support the LPs and support the whole cannabis industry using scientific facts and methods. And we should never disregard them for business or profit. What is seen and is escalating, has been escalating in the past 6 months in Canada, is that unfortunately, regarding different methodologies and different technologies and methods to test for microbial contamination, each lab is trying to announce and promote that only the way that they test for microbial contamination is accurate and everything else is unacceptable and unreliable. Qpcr has specifically been under attack in the past few months, as here are only a few labs who can afford it or who could validate their methods. Again, I'm just using it as an example. Some labs were saying that to test for microbial contamination, Qpcr is not approved and acceptable by Health Canada to test cannabis, which is absolutely false. And we have Health Canada's approval since January 2020. That as long as your method is validated, you can use Qpcr to test for microbial contamination in cannabis or cannabis-derived products. There has been some false information or flawed studies, published, questioning the accuracy of Qpcr results. Qpcr is a more accurate technique because it relies on DNA amplification rather than subjective interpretation of a microbiologist of the color that they see. Also, since the identification of contamination is at DNA level, the possibility of mistaking one organism for a similar one is almost non-existent, which is far more specific and accurate. And Jason, look, now after the pandemic and the COVID tests and everything, I think no one should doubt the reliability of the advanced technology of PCR. Look at what has been used here. Also, there's been some questions in the market about Qpcr reporting false positives because it reports not only live, but also dead bacteria. This is not correct either. If you know and use the new kits and new technologies available, which differentiates and separates live and dead organisms, then it won't happen again. So just for example, here at Sigma, we test many dried flower samples, which are from an irradiated batch. And we have never, ever had an issue with Qpcr reporting the dead bacteria and falsely staining the samples.
Mhm.
So the point that I'm making here is that Health Canada has given this flexibility to pick between different standards, different methodologies, and different technologies.
Right.
I think this is the responsibility of the labs to simply focus on the scientific facts and educate the market, let them know the good, the bad, and the ugly about each and every one of them,
Uh-huh.
and let the client clearly know about their options. No, there's not only one option that works, one option that's accurate, one option that's acceptable by Health Canada. No, there is more than one option. Look at different factors and pick the one that better works for you. And again, there are many studies and publications, including even one from USP. Since cannabis is not federally legalized in the US, USP cannot just publish a monogram on these tests, but there has been peer-reviewed papers and articles declaring Qpcr is the most reliable and accurate method for testing microbial contamination in cannabis products. Specifically, I'm talking about cannabis. Of course, FDA and AOAC have hundreds of accredited methods based on PCR. But the good news is that good things are happening, not necessarily here in Canada, not necessarily under Health Canada, but if you look at USP, if you look at AOAC, if you look at the recent announcements and publications, you can see that, for example, AOAC recently approved the Qpcr assay for testing aspergillus in cannabis, and also E. coli, salmonella, and total yeast and mold are also in process. So we need to take a look at the real science behind it and underneath it, and based on that, with the lab's advice, based on the science of it, of course, the LPs have different options to pick from.
That's right. Yeah. I mean, and really the key thing was what you said in the beginning, it's validated, right? I mean, as long as the method and procedure is validated, it doesn't really matter the level of technology. And, we understand that Health Canada doesn't want to exclude certain labs just because a PCR is not an inexpensive piece of instrument for any lab. And it's been around for a number of years in the biopharma world. And, now we're seeing it trickling into other uses, cannabis now. So have you been hearing any sort of, this kind of circles back to what we talked about earlier, have you been hearing any testing standards or guidelines from Health Canada being released anytime soon, whether it's for microbial or any other tests?
About microbial, they have done a lot in some other fields and tests recently, but I'm not aware of anything in the pipeline for microbial contamination specifically. Mm-hmm. Even the approval that we received from Health Canada about Qpcr for testing cannabis, it was in response to an email that we sent them, not a public announcement here. Mm-hmm. But Health Canada and regulations aside, I think, again, it's a very good idea to keep an eye on USP and AOAC and see what they are doing regarding cannabis. Mm-hmm. About the other fields, I personally believe that Health Canada has done and is still doing a lot, especially recently, especially in the last year, in the last few months, to bring more clarity to the market. But mm-hmm. Of course, there is still more left to do. There has been some clarification, late last year, for example, about testing heavy metal specifications based mm-hmm. on the intended use. And, we saw some changes in the pesticides limits. However, there are still some gray areas. There's still some, vague areas. For example, pesticide specifications for oil is still causing confusion, as the new regulations of March 2021 don't recognize oil anymore in the list and limits and table, and the previous specifications are not valid anymore. But the column for oil is still there, so it causes some confusion. And the manufacturer should come up with their own specs, which shouldn't exceed the highest value in the pesticide table. Uh-huh. So about residual solvents, for example, we hear this from our clients that, okay, there is a list and there are limits by Health Canada. Clear, we see that. But should they test for each and every solvent listed there, or should they only test for the ones that they have used? Yeah. For microbiology, just discussed it, that there are different pharmacopoeia that Health Canada recognizes. Uh-huh. I think something really good that happened just very recently, a few weeks ago, was Health Canada clarified that for potency, for cannabinoid content, static reporting is not acceptable. They made it very clear that for each batch, you should just test one sample, and mm-hmm. that's what should go on your label. Basically, that's what your label claim should be. So you cannot simply say that historically, we have seen that this strain has this much THC and this much CBD.
Right.
which wasn't reasonable at all, because we have two very different concepts here. You have strains, but you have also cultivars, because the cannabinoid content is not only based on the genetics, it's not only based on the seed. The environmental factors also affect it, from the nutrients to light to temperature, everything. So you cannot assume that each and every harvest of the same strain will give you exactly the same cannabinoid content. So I see it as a good step forward, and I appreciate what Health Canada did. They very, very clearly said that static reporting is not acceptable. Each and every batch should be tested. And this is a good step forward. Another field that I can think of is that, unlike some other jurisdictions, Health Canada's regulations has been absolutely silent about something like batch size. An LP is absolutely free to pick any batch size that they want, from 2 kilograms to 2,000 kilograms. And there's also nothing very clear and firm about sampling procedures. So there are more steps that Health Canada can take, but I'm happy with what we have seen in the past few months, and what they are doing to clarify it more.
Yeah, and that's you're absolutely right. Given that we're the first, sort of G8 nation to have a legalized system, you would think Health Canada have a lot of these regulations in place. And even with sampling, I know in their guidelines they refer you to sort of the herbal sampling procedure in EP, and it's still not very clear for whether it's the same for cannabis or how that could be applied. So what I guess based on your opinion, right? Why do you think that the pace of some of these guidance and guidelines are being rolled out by Health Canada, it's coming out so slowly, when there's still so much I guess gray area within this field of analytical testing? Why is it taking so long? Is it just because of resources, or they have bigger problems to deal with? What is it that is causing such a slow rate of release for some of these guidances?
When it all got started, with legalization in October 2018, it was understandable, it was acceptable that they wanted to get something started, and some flaws, of course, would exist when you do this. After that, we saw a huge backlog, huge pressure on Health Canada regarding licensing. There was a very, very long queue for license applicants, especially for cultivation or processing, even for analytical testing. So my understanding is that during the next year, they were very busy taking care of that backlog. Then in October 2019, there was a second wave, Cannabis 2.0 products, which was a whole new field, and they had to just take care of that and put things in place. I think now, now in mid-2021, or my expectation was actually in late 2020, they should have been able to take care of that and focusing more and more on what's missing or what is not clear. Again, let me express it once more, that in the past year or so, or in the past 6 months, we have seen some good steps forward from Health Canada, and now we see more clarity. I gave you a few examples of it. But I agree with you. I think they can do better and they can do faster, because that's a serious need in the market for more clarity.
Yeah, no, and speaking of, Cannabis 2.0 and talking about clear guidelines, one thing that they've been very clear about from the start for analytical testing are validating test methods, which we touched on a couple times already. And with Cannabis 2.0, came a new set of products, or new classes of products, and edibles, beverages, topicals, right? The need of validation, especially for those products. Can you talk about some of the challenges and complexities, dealing with some of those sample matrices?
That's a great question and great point, because this is clearly required by Health Canada, that cannabis and derived products need to be tested and reported using validated methods. This is very clear. The purpose of validation is to establish some characteristic parameters of the method in, quantitating the analyte. If a lab has developed a method to test for a contaminant or a profile and cover all analytes, it's straightforward for basic cannabis products such as flower, distillate, and isolate. I mean, if you have validated your method for dried flower, every dried flower is the same. You don't need to go validate it again, and you can just use it to test any dried flower samples that you receive. However, Cannabis 2.0 products have an additional level of complexity, because of variety of the compounds in their matrix. So you cannot simply have a validated method for all cannabis topicals.
Right.
As you know, what is in a cannabis water-based gel is different than a cannabis oil-based cream.
That's right.
And even two creams have different formulation and different ingredients, and they are not completely similar. So this is something that Health Canada has started investigating in auditing LPs and their certificates of analysis, and will most likely expand the scope and depth of the investigation. Again, as I told you, this is a personal experience. In the past few months, it has happened more than once that a client of us has been audited by Health Canada, and Health Canada asked them to go to the lab and provide and get the validation reports and provide them to Health Canada, which is good news. Actually, this is what has to be done. So this is what is happening, but there are still some misconceptions in the market in this regard. And for example, some labs assume that when you have a method validated for an edible, for example, it would, after all, be edible, and you instead of a validation, you can simply do some spike recovery, which is inadequate, which is risky, and doesn't exactly fall under what Health Canada is asking for, which is validation. Right. This approach would not demonstrate the efficacy of sample preparation process in quantitatively extract the analytes from various complex sample matrices. So this can lead to incorrectly reported results, resulting in safety and efficacy issues for consumers, risking recalls and both financial and reputational damage to the LP. So on one hand, I think we go back to what we discussed in the previous question. Yes, Health Canada needs to bring more clarity to the market, but meanwhile, it's the responsibility of the labs to do the right thing and to do the complete thing. And also it's the responsibility of the LPs to make sure that they work with the right lab who does it in the right way.
Yeah, no, that's those are all great points. And I know we're talking about a lot about validation. So for those of you in the audience who might be wondering, what it actually entails, essentially the lab needs to demonstrate that the method is accurate, it's precise, that you have a linear range, right, that we determine. And then with edibles and topicals, there's a lot of other ingredients that could produce interferences with your signals. So these are things that contract labs will definitely have to look up for. And this is trying to explain this to some of our clients too. They're like, well, they have validated methods on edibles, but it's not your particular product, right? I mean, so that's great. And one of the things that we're starting to hear a bit more and more in the industry as well is cleaning validation. And cleaning validation for those who are a little bit new to this, but basically it's to ensure that your cleaning practices and procedures are sufficient, and you take swab analysis samples of surfaces, and you send it off to the lab for testing. So I just want to know from your perspective, Ashton, have you been seeing more and more requests for cleaning validation samples, and why is this important for the industry?
This is another important point and a great question, as this is another field which Health Canada hasn't yet got really serious about implementing, but it should not be far away, I expect. Again, there's a misconception in the market that cleaning validation is only required under GMP. And so we are mostly seeing a demand for this by LPs who work under GMP. However, per Section 517 of GPP, the effectiveness of the sanitation program should be monitored and verified. So all LPs should take cleaning validation seriously. It's not only under GMP. And as you said, I'm just trying to add an additional layer of clarification here. The purpose of cleaning validation is to demonstrate that residues that can cause product contamination are removed effectively from the equipment through the established cleaning procedures. And they are not being introduced to the next lots, to the next batches. This could be, residue of active ingredients that they should be careful about, even the cannabinoids. Because what if, for example, you were just processing a high THC batch, and now as the next batch, you want to do a CBD only THC free batch. So when we say contaminations here, in the world of cleaning validation, it could even be your cannabinoids, it could even be your APIs. Also bioburden, microbial contaminations, that's another part of cleaning validation. And even the detergent that you use for cleaning, you need to make sure that no residue is left and no residue is transferred and introduced to the next batch. So this is done, again, as you said, by testing samples collected either directly from the surface by swab testing or indirectly through ring sampling. I hope, I expect that very soon Health Canada will bring more clarification to the market about this and make it clearer about how necessary and how important it is. But I think, regulations aside, GPP and GMP requirements aside, I think if an LP really cares about their consumers, if they care about the efficacy and safety of their products, they should take cleaning validation more seriously.
Yeah, no, absolutely. And this has been around for food and pharma, and it's not a new concept. But we're hearing more and more in the industry. So that's glad that we're glad to know that it's sort of steering towards that direction. And in terms of, I guess, speaking of contamination that that you spoke of when it comes to microbials, one that they kind of tend to forget, and we're also reading a lot of stories of this from labs in California and elsewhere, that there are some viral contamination that's going around, that's causing a lot of headaches for licensed producers. So what's happening in that field, and have you been sort of researching, investigating into analysis for viral contaminants?
Yes, growers worldwide are seeing cannabis disease symptoms caused by viruses, and the viruses have been detected in cannabis and hemp crops in Israel, California, Colorado. We should not assume that we are safe here in Canada because we mostly grow indoor. Let's say some of these viruses are most commonly spread, via infected pruning tools, and also there are unofficial reports of them being already being found in the crops here in Canada. The most common virus among them is House latent viroid, or HLVD, which won't kill the plant or even cause obvious symptoms of infection. You don't see any curling or yellow leaves. However, plants will show subtle symptoms. HLVD will greatly reduce the quality and quantity of the flower the infected plant produces. And during the vegetative stage, plants will grow shorter with smaller leaves and tighter node spacing. So flowering plants will have a smaller, looser buds with much fewer trichomes. So symptoms include stunted trichome production, lower yield, and perhaps more importantly, reduced potency. So this is serious. This is really serious, and the producers should take it seriously. What we do here at Sigma is that our lab screens cannabis plants for 3 common virus infections: HLVD, as I already mentioned, lettuce sclerosis virus, and also cannabis cryptic virus. And the 2 pieces of good news that I have is that, number one, we recently are seeing more and more demand in the market for it. So it seems like the growers got the message, they got serious about it, and more and more of them are checking their crops, before harvest for viral contaminations and send the samples to us to be tested. And the other good news that I have to share is that, of course, we do not cover the whole Canadian market, but among the LPs that are working with us, among the ones which are sending their samples to us, their leaf and flower samples to us to test for those viral contaminations, so far we have not seen any case. So even if it exists in the Canadian market, at least it's not widely spread.
Okay, that's good to know. And correct me if I'm wrong. I mean, Health Canada, they have somewhat guidelines. We discussed on microbials, but do they have guidelines on viral contaminations at this point, or is it still relatively new for them?
No, what Health Canada has been focusing on is that they have been trying to assure the safety and efficacy of the cannabis products for the consumers. This is something that won't affect the consumer. What's the worst case scenario? Let's say that you have a cannabis plant and it's contaminated by one of those viruses, and someone consumes them. There is no harm, no damage to the consumer. However, it would affect the crop and the harvest. So since it has not been falling under that umbrella, so far Health Canada has not become serious about it. There are no regulations, no requirements in place. And since it would just affect and damage the growers, this is what they should be careful about.
Right. I mean, you talked about yields, you talked about quality. I mean, it impacts your bottom line. I think that's
cannabinoid content, yes.
Yeah, your bottom line, your profits, right? I mean, that's probably what most LP would probably want to hear about. So that's great. And the testing is done PCR, or are there other techniques to screen for those 3 viruses?
No, also on Qpcr, because Qpcr uses that DNA amplification, it could be used both for bacteria and virus.
Okay, fantastic. Okay, so that's great. And speaking of, and we mentioned GMP a little bit earlier on, right? And earlier in this conference, we also had a panel on UGMP as well. For those who are looking to get their GMP certification if they want to explore into the European market. Is there any impact, with labs like Sigma, or does it impact any of the processes that you have with your clients? How does that work?
Yeah, that's an important point, and we take pride in being the very first cannabis-focused GMP-certified lab in Canada, because we expected that and we saw the need in the market. As you know, still GMP is not mandatory here, but that's a very important point here. Not only domestically, not only in our market, but also there are many LPs here in Canada who have international operations or who do export to Europe or to Australia or to other jurisdictions where GMP is required and mandatory. So again, as you said, there's another panel specifically about GMP, so I'm trying to be brief here and only clarify a few misconceptions here. There is a big difference between production and testing under GMP. There are some mutual recognition agreements about GMP in place. One is between Canada and European Union, and it's not something new. It goes back to 1998, but has been updated a few times since. So under this agreement, GMP compliance issued in each jurisdiction is found equivalent in the other one. So basically, if there is a lab in Canada, a cannabis lab in Canada who is GMP certified and basically has what Health Canada called drug establishment license, that would be enough and acceptable in even European Union, even in Australia, as a GMP certification. Again, I'm only focusing on testing and lab. There are some differences between testing and production. One of them that you mentioned, because you mentioned EU GMP, I understand that it applies to manufacturing. I understand that you need to, get audited and certified by a body in Europe if you want to export to Germany or another European country. But again, let me express and clarify and emphasize that there is no such thing for testing. There is no EU GMP lab. The term EU GMP doesn't apply to testing. There is only GMP.
Right.
So that's something that should be taken into consideration. And another piece of information that I hope the audience will find useful here is that if, for example, you're targeting a European market and you want to export there are two different sides to it. One is the requirement for GMP. So of course, if you want to give your buyer, the importer, a certificate of analysis, it should have been tested and issued by a GMP certified lab. But there's another side to it too. GMP aside, there are different testing requirements. For example, the list and the action limits for pesticides in Europe is very different than the list of 96 pesticides that here we have in Canada. Right. So if someone is serious about expanding their business, expanding their operation, or exporting to Europe, they should see this side of it too, and not only the GMP certification.
Okay. Well, that was great. I mean, that was insightful. And I think what we'll do, Ashton, is those are kind of my burning questions I had for you, but I know the audience probably have quite a bit of questions for you as well on this topic. So we're going to end it right there. Ashton, thank you for really your wonderful insight into the world of analytical testing from the lab's perspective. There was a lot of great information that we covered. I want to thank everyone who tuned into this panel as well. And before we dive into the Q&A, where can the audience find you? How can they contact you? Is there any information that you want to put out there?
It was my pleasure. I look to the QA session and hopefully I'll be able to share some more information there. Yeah, we'd be happy to help if anybody has any question. As I said, at Sigma Analytical, we're not only a typical testing lab. We offer different types of services, including consultation, and even the services aside. If anyone has any question, if anyone needs any clarification on testing requirements or the regulations or testing methods, we'd be more than happy to help. You can reach us either on social media at Sigma Analytical is the handle, or website sigmaanalytical.com, and we'll be more than happy to help.
Fantastic. Well, we'll see you in the Q&A right now.